All right, in this video, we're going to talk about another type of buffer system that exists in the body. And this one in particular is going to be something known as the protein buffer system. And so when we say this is a protein buffer system, what we're talking about is specifically the amino acids that compose our various proteins. So we know that all proteins are made up of a string of amino acids. So if we draw an amino acid like this, each of these circles is indicative of a single amino acid, and they are formed into this one large protein or a polypeptide chain, right? And we call it polypeptide, poly meaning many, and peptide referring to the peptide bonds that bind each of these amino acids together. So if we look closely, this right here, this is a peptide bond, this is a peptide bond right here, here, and so on and so forth for the entire protein or the entire polypeptide chain. Now, I robbed this image off of Google, but what this image is showing us is a structure of an amino acid, and what happens whenever you put these amino acids together. What you see is you see this end here, which is a carboxyl end. Carboxyl is COOH here, and then this here we have an amino group, which is NH2, right? And so what you happen is the carboxyl of one end of an amino acid and the amino group or the amine group of another will combine. And when that happens, you lose, let's change to a different shade of blue here, you lose water, right? So 2 hydrogens, 1 oxygen, right? And so whenever we lose that, we lose water. So this is a dehydration synthesis reaction. Dehydration synthesis reaction. We are losing water in order to synthesize something. So water is going to come out, and you can follow the remaining atoms, the remaining elements here. So here's a carbon, here's that carbon, here's an oxygen, here's the oxygen. Here is a hydrogen, here's that hydrogen, here's a nitrogen, here's that nitrogen right there. So you can see the only things we lost were these 2 hydrogens and this oxygen in the form of water, and that's why this is a dehydration synthesis reaction. But that's not the main focus of this video. The main focus is on the nature of our proteins and our amino acids and how they regulate our pH. So again, let me write, this is our protein buffer system, right? And specifically, we're talking about our amino acids, okay? So what we see is on one end, we have the amine group, which is this amine group, and on the other end, we have a carboxyl. Now, you can think of the carboxyl group as being slightly acidic, and you can think of the amino group or the amine group being slightly basic or alkaline. Okay, slightly alkaline. Now, every protein or every large polypeptide chain on one end will have an amino group, the amine group. On the other end will have the carboxyl group. So here, as you see on this small dipeptide here with 2 amino acids, here you have an amino group, NH2. Here you have a carboxyl group. It would be the same for this long polypeptide here. So say on this side down here you have NH2. On this end over here you have your carboxyl, okay? Now, each end can function as a buffer depending on the state of the environment that this protein is in, right? So I said that if you're talking about the carboxyl end, you're slightly acidic, and if you're talking about the amino end, you're slightly basic. So imagine we get put into a solution that is slightly basic, right? Or if we're talking about our blood, remember our blood's pH is somewhere generally between 7.35 to 7.45. Say we're put into a solution and we become too basic. So say we come up here and now we're 7.5 0.52. Okay. What can happen, and whenever we have this increased pH, right? Whenever we have this increased pH, we have decreased hydrogen ion concentration, right? So what we need to do to fix this and get us back to this homeostatic range here is to increase the hydrogen ion concentration. And one way you can do that is when you look at this carboxyl end, this, let me erase this real quick, this individual hydrogen here can become free, and now you have a free-floating hydrogen, right? You can imagine if you have lots and lots of proteins, this could happen many times, and you can increase the hydrogen ion concentration. So now we've increased hydrogen ion concentration, right? And that indicates a decreased pH, which indicates an increased acid level, right? And we're dropping our pH, and that is opposite of where we started. So we were too high, now we're back, and say maybe now we're 7.38, and we're now homeostatic, right? So now let's say this happens, but the opposite thing happens. So let me erase all this. Say now we're at a pH of 7.27, okay? So now we're too acidic. So that means what we have is we have too many hydrogen ions, okay? So we have too many hydrogen ions. So let's go back over here and say we have too many. So we have lots and lots of hydrogen ions free floating in solution that is causing our pH to drop and our acid level to increase. Well, what can happen on this amino end is now you can take one of these hydrogens and cause it to bind to this amino group, and now you've formed ammonia. So now you've gone from NH2 to NH3. So you've gone from 2 hydrogens to 3 hydrogens. So you added a hydrogen to this functional group here, and what that did is thus reduced one of the hydrogens. Okay. Now, if you had lots of proteins, obviously you could begin reducing lots of these hydrogens, and thus buffering the pH of your solution. So we were too low, right? So this was too low of a pH to begin with, right? Or too high of a hydrogen ion concentration, right? And by buffering this, we've thus taken up some of these hydrogen ions. So now we've gone from this to decreasing our hydrogen ion concentration, which is what we wanted because we had too many originally up here, right? And whenever we did that, we thus increased our pH, and thus we decreased our acidity level, right? So we've gone from 7.27, say now we went back up to 7.41, right? So now again, we're in that nice homeostatic range, okay? And again, this here was opposite of what we started with here. And that's the whole idea. We are resisting, whenever we talk about any type of buffer, we're resisting significant changes in pH, right? So we may change a little bit, but this buffer is going to prevent us from having significant changes. And our protein buffer system is one example because we have a carboxyl end on one end that's acting slightly acidic to donate hydrogens, and then we have this amino group here which is acting slightly basic to bind hydrogens to it and thus reduce their concentration in solution, thus bringing up the pH as we saw right here, and thus bringing our pH and our acid level of our blood